Okay, so here we're back with our data table, and I calculated the numbers here on the bottom for you. So you should compare that to your own results. For me, the sum of the male heights came out to 2,838 centimeters, and the average for the males came out to be 177.4. The sigma is 8.18. And for the female side, it came out, the sum was 2,667, and the average female height 166.7, and the sigma 7.67. So if you were to try to put this into some sort of a graph, then it would look like this. So here's a bar graph representing male versus female height. The height of the bar, so here are the males. The height of the bar is the average. So we have an average male height of 177.4 centimeters. I rounded it to the full number, so it'll make it a little bit easier since you can't really see it on the graph anyway in fractions or decimals. So anyway, so 177 plus minus 16, so that's 1.96 times sigma. So it's almost 2 times the 8 right here, so about 16 centimeters. So 177 is the average height plus minus 16 centimeters to get the 95% confidence limits. And for the females, the average height is 167 centimeters approximately, plus minus 15, so that's where we get 1.96, so almost 2 times sigma is about 15 centimeters. So 167 plus minus 15 centimeters to account for the variation in our sample, in our group. Now, when you're looking at this, if you eyeball it and you just think about it, so is there a significant difference, yes or no? Well, it's hard to tell because there's so much overlap and really the heights aren't that different. So we have to do the math to see if there is a significant difference between these two groups with 95% confidence. And so for that, we need to do all of our calculations. I put the table, filled out the table, the numbers down here. You can take a look at that. So all of the numbers just transferred from the table above, and then you have to put in your numbers into your T calculation. Here's just the formula, and then here are the numbers in there. So we're subtracting male minus female average height, and these sidebars here, that means the absolute value. So that means we don't want to have a negative number here. We just want the absolute difference between male and female height. In this case, it comes out to be a positive number, so that's fine. So it's 10.7 centimeters on average difference. And the bottom here, the numbers filled in, just transferred from the table. This whole term here comes out to be 62.84 times in 1/16 plus 1/16, so 1 over n1, 1 over n2 comes out to be 0.125. So here are your 10.7 centimeters absolute difference between male and female height. Then this term here comes out, that's quite simplified now, to 2.8. So our T calculated value is 3.82. And we're going to put this down here. Calculated T is 3.82. Our degree of freedom was 15, which in the T table gives us a cutoff value of 2.13. 3.82 is bigger than 2.13, so our answer is yes, there is a significant difference between the two groups with 95% certainty, so, and p.05. So that means we reject the null hypothesis and we come to the conclusion that yes, there is a significant difference, and our initial hypothesis that there might be a significant difference turns out to be true with 95% confidence. Okay, I hope you got something out of this here. Please try to reconstruct the numbers. I try to make it as obvious as possible. Now, as far as testing is concerned, I won't give you any kind of quiz questions where you have to calculate a t-test, but you should know what a t-test is for. So when you think about it, why do we do a t-test? Well, we're comparing two groups. So we're forming the averages, and then we look at the how tight the data set is, or how much dispersion there is, how much variation there's in the sample. We take all of that into consideration when we're comparing these two means, and then our t-test calculation gives us the answer whether there is or is not a significant difference between the two groups. So two main takeaways from all of this. Why do we do a t-test? Because we're comparing two groups of data, and we want to find out whether there is or is not a significant difference. And I want you to use these terms. That's really this statistical terminology. That's something that you just need to use these terms, significant difference, and everybody knows what you're talking about. Okay, and finishing up this lab here with taking a look at this experiment B that you don't get to do because we're not in an actual physical lab, and I don't really have agar cubes that I could provide for you to do this experiment. But here's the point of this experiment. In the actual lab setting, you're given agar cubes of different sizes. And the idea here is to demonstrate how important cell size is so that you can take advantage of diffusion. Diffusion is a process where materials run down their concentration gradient. It's a passive transport process that does not require energy, and so therefore cells absolutely love it. They like to take advantage of concentration gradients that exist in nature, such as, for example, the oxygen concentration in your blood versus the oxygen concentration in your air that you breathe. And so you have these concentration gradients everywhere, and you want to take advantage of diffusion because it's energetically free. And the only problem that you have is that diffusion, there are some parameters, and materials don't diffuse that fast. That means they can't overcome huge distances. So if a cell wants to be able to get rid of waste products or take in nutrients by taking advantage of concentration gradients and utilizing diffusion mechanisms, then we need to have small diffusion distances. And that means cells better be small, otherwise it would take forever to provide like nutrition to the center of the cell. So these agar cubes, they are placed in a vinegar solution, which changes the pH, and it changes the color of the cube, and in this way you can monitor the diffusion. By the end of the experiment, you can see that the biggest cube, there's hardly any change in color because the volume is so huge that the vinegar didn't reach the center in the time given. We're running this experiment for 10 minutes usually, and the smallest cube is almost entirely changed color because it's small, and the vinegar was reaching the volume of almost the entire volume of the cube within the time given. So anyway, since you didn't get to do this experiment, we don't do this graph, so on number 3, I sort of gave you a hint on why the diffusion medium changes the color. It's because we're putting a pH indicator in there, and vinegar is an acid, and so it changes the pH of the agar cube, and that changes the color because the pH indicator shows the change in the pH. Number 4, if a cube represented a living cell and the diffusion medium is a substance needed to survive, what problem might exist for the largest cell? And again, this is the problem. If you have a large diffusion distance to overcome, then it would take forever to reach the center of the cell. Imagine if your red blood cells were the size of pennies, for example, then how long would it take for oxygen to go all the way to the center of this very large size cell? It would take way too long. You don't have that much time to zoom by the lungs and pick up some oxygen. So cells better be small to take advantage of diffusion mechanisms. Number 5, describe the relationship between cube size and the rate of diffusion. The rate of diffusion does not depend on the cube size, so that's a constant. The rate of diffusion depends on things like temperature. The more energy is in a system, the more collisions you get between molecules, and that will change the rate of diffusion. So what you could do, and I'm going to suggest that same experiment for next week, you can take some hot water and some cold water in a cup, maybe a glass of water, and then put a sugar cube into the cold water and into the hot water, and watch what happens. You should see that in the hot water, and it's the same substance, but you will see that in the hot water, the sugar cube, even without stirring, it will diffuse much faster than in the cold water. And the reason for that is that heat is a form of energy, and energy means collisions between molecules, and that means that as these molecules are colliding with one another, you just get more mixing, or in this case, diffusion of substances. So number 6 described the relationship between cube size and the extent of diffusion. So how much as percent of the volume got diffused. And of course, the smaller the cube size is, the larger the extent of diffusion given a certain amount of time. On 7, so describe what happened to the following cells as the cells grow larger. So here, well, the surface area, if an object gets larger, of course the surface area will increase. There's no doubt about that. The volume, as something gets bigger, volume will also increase, but the surface area to volume ratio will decrease. So let me give you an example. Let's say the surface area of a 3 centimeter cube. So that would end up being 54 centimeters squared. So that's 3 times 3 times 6 sides. By contrast, a 1 centimeter cube is 1 times 1 times 6. So that's 6 centimeters squared. Sorry. And the volume of a 3 centimeter cube is 3 times 3 times 3. So that's 27 centimeters cube now, and a 1 centimeter cube would be 1 times 1 times 1, and that's still 1 centimeter cubed. Now let's take a look at what happens to the ratio. So here our 3 centimeter cube has a surface area to volume ratio of 2. So 54 divided by 27 is equal to 2. Now the surface area to volume ratio of a 1 centimeter cube is 6 divided by 1, and that's equal to 6. So you can see that as the cube gets bigger, so we're going from 1 centimeter cube to 3 centimeter cube, we're comparing the surface area to volume ratio change. As the cube gets larger, the surface area to volume ratio decreases. And so on 8, we can now answer, according to the results of your investigation, which cell size and surface area to volume ratio best meet the diffusion needs of living cells. And basically the rule here is the smaller the better. The smaller the volume inside of the cell and the larger the surface area, of course within limits, but that's the reason why cells are small, because they want a small volume to service with a large surface area. Now if we calculate on 9, the surface size of some human cells is 0.001 centimeters. Using the formulas in this activity, calculate surface area to volume ratio, assuming the cell is a 0.001 centimeter cube. Describe the extended rate of diffusion into this living cell as compared to the smallest agar cube. So I did the math here for you. So the surface area of such a cube with the 0.001 centimeter side length will be 0.001 squared times 6 sides of a cube, and that's this number right here. And the volume of such a cube with the side lengths will be 0.001 to the third power, so cubed, and that's this number right here. If you take the ratio of 0.0000006 divided by 0.000000001, if you take this ratio, then you will find that the surface area to volume ratio of this little cube is 6,000, and compare that to our these cubes, these fairly large 3 centimeter and 1 centimeter cubes. You can see here still the 1 centimeter cube already has a surface area to volume ratio of 6, but this tiny little cube that is realistically about the size of a cell has a surface area to volume ratio of 6,000, which indicates that we have a huge surface area compared to a tiny volume, and that's what you want. Then this way it's easy for cells to take advantage of diffusion. It's also easy to communicate. It's easy to embed a lot of transporters and channels and receptors into a large surface area, and then service a relatively small volume or interior of the cell. And we will continue this discussion in the next lab. Now you do have a quiz that will be covering labs 1 and 2. Let me give you some tips on how to study for this quiz. It will be worth 20 points. You have 20 minutes to complete the quiz, and it will cover quite a bit of metrics problems, so you should have a simple calculator handy. It will also go over some of the what we discussed about the scientific method, what's a good hypothesis, what's that is testable, for example. Then you should also know some of the statistical terminology, mean, mode, and range, and whatnot. And you should also know why we do a t-test, and what the result is that we, or the meaning that we get out of a t-test, whether there is a significant difference, yes or no, and you should be just fine.